purpose of this viral cultivation will be used to identify the virus in the clinical samples to do the research. And method of virus cultivation will be helpful for inoculation of virus to the animals, inoculation to the embryo egg, and tissue culture. Animal inoculation which are not cultivated in embryo eggs, tissue culture and cultivated in laboratory animals such as mice, guinea pig, hamster and rabbits. So the selected animals should be very healthy and free from the communicable disease. And mice are the most commonly used model, and mice are susceptible to typovirus and some other virus to inoculate into the intracellular and intranasal group. Inoculation of the virus is very important role, essential for the particular viral studies, and live animals like monkeys, mice, rabbits, guinea pigs, parrots, and widely we can able to use different type of animals. So here the advantage of animal inoculation will be diagnosis and prognosis of clinical symptoms are determined, production of antibodies can be identified, primary especially certain viruses. Mice provide reliable model for studying viral replication, used for studying immune responses, epidemiology and non-infectious. Disadvantages of this animal inoculation is expensive and difficulties are in maintenance of animals, difficulty in choosing of animals for a particular virus. Some human virus cannot be grown in animals, or can grow but do not cause any disease. Mice do not provide models for vaccine development, so that's why we need to go for alternative. So another type of method will be embryonic egg. Good passer is a person who identified the first time this method of embryonic egg cultivation. So the embryonic egg depends upon the type of egg which we are using. So virus inoculation embryo, 7 to 12 days old embryos. So for inoculation, eggs first prepared with the cultivation to the cell surface of the first disinfected with the iodine and patented with a particular small sterile needle, and we can transfer into it. So after the inoculation, we can incubate it 37 degrees Celsius, 36 degrees Celsius for 2 to 3 days. After the growth of virus, we can able to identify and we can go for further studies. So different type of membranes are there: chorial type membrane, allantoin cavity, amniotic sac, yolk sac inoculation. So advantage of these inoculation the embryonic is widely used and is ideal substrate for viral growth and replication. Isolation and cultivation mainly avian and few mammalian virus. Cost effective and maintenance is much easier. Less labor is needed. Embryonics are readily available. They are free from contaminating bacteria and many viruses. Disadvantage of this embryonic inoculation is the site of inoculation for various viruses with different virus. That is, each virus have different sites for their growth and replication. So next method is cell culture. The tissue culture classification to organ culture, experiment culture and cell culture. So here we need to pick up each steps like monomer incorporation, clonal cell incorporation and infection into the virus. So organ culture, experiment culture is very easy compared to the cell culture, and finally we can be subdivided into the types of cell culture will be 3 types: primary cell culture, diploid cell culture and secondary cell culture. So the primary cell culture will be very easy, like we have to take a particular cell line and we have to allow to grow the particular virus and we can go for further studies. Diploid cell culture is single type, the original diploid chromosomes, so subdivision for 50 times. So with viral vaccine production that we can be adapted to this kind of diploid cell lines. Currently cell line is helpful for growth in various type of cells like cancer cell line studies that we can do with these kind of cell lines. Example like HeLa, HEP2 and KB cell lines. So advantages of this studies will be more reliable and helpful, easy to do the virus cultivation for this various type of viruses. Thank you.